Good afternoon, everyone. I'm Colin Duckett, Vice Dean for Basic Science, and I'd like to welcome you all to the School of Medicine's second annual Research Week program and our first ever totally virtual Research Week. We're in for an exciting week-long celebration of basic translational and clinical research, and I'm really glad you could join us. Before we start with today's presentations, I want to briefly mention the other superb lectures that we can look forward to this week. I hope you can join us for all of these talks. We've invited a number of exceptional researchers and have chosen a wide range of fields that we think will be of interest to you. I encourage you to grab your lunch, tune into all of the presentations and enjoy, even if they're not in your direct field. So today, you'll hear presentations from three truly outstanding Duke faculty members. First, we're going to hear from Dr. Kaf Deraza. Kaf will be talking about his work to unravel the molecular basis of emotions through the analysis of local field potentials in the brain. Kaf's going to be followed by Dr. Claire Smith, assistant professor in the Department of Molecular Genetics and Microbiology, who will be sharing some of her work studying the role of the genetic background of both the host and the microbe in contributing to the dynamic responses to infection. And finally, Dr. Ed Miao, professor of immunology, will describe his work dissecting out the suicide programs that are triggered by host cells in response to bacterial infection. So without further delay, let's get started with Dr. Kaf Deraza. Dr. Deraza is the K. Ramakrishnan Associate Professor in Psychiatry, and he also holds positions in appointments in the departments of neurosurgery and neurobiology. The title of Kaf's talk is Mapping Emotions Towards Precision Psychiatry.
All right, so there have been a tremendous number of lessons of 2020, and the learning lesson for today is don't make fun of cardiologists. All right. So again, jumping in where I left off, this is the heart, and if one was to start having problems with your chest or your heart, you go in the emergency room and one might put leads onto your chest and measure electrical patterns through the heart. And we're able to do this. When we do this, we come up with a set of normal patterns, which we call the electrocardiogram. And this gives an indication of how a given heart is working. And this is particularly important because individuals can come in which they have changes of these rhythms in the heart, which are diagnostic of disease. And so psychiatry has nothing like this that is both gives you information about an individual's range of normal function as well as gives you an indication of disease function as well. And in order to achieve that, there's several key features of the electrocardiogram that we'll want to build in. First, the map that is generated by the technology should decode new subjects. And what that ultimately means is that if a new subject or new individual comes in the emergency room, it should tell us something about that person's heart. Many of the tools that have been developed in the neuroscience enterprise, they may tell us something about the same heart that were used to learn the model. In other words, if I learned it on your heart, I would know that you were running up a flight of stairs, but it wouldn't necessarily go to the next heart. Secondly, it should be linked to interpretable biology. In other words, there are features in the EKG that can tell you when you have a clot in your LAD or whether parts of your heart have lost their ability to move blood into them. And then it should go to new contexts. In other words, it should tell you about normal function. It should tell you about when you're on a treadmill. It should tell you about when you have broader heart disease like pericarditis as well. And so the idea is to get to precision diagnostics, any diagnostic should achieve all of this. Now, the challenge is obviously much greater in thinking about this in the context of psychiatry than it is in terms of cardiology, because it's hard to think about preclinical models. So as I've put up here, and this is a bit done in a bit in jest, because animals certainly do have facial expressions that give indications about what we would call emotional states. When we think about things that bring in the individuals in the clinic, whether that's guilt or homicidality or suicidality, we don't do that very well at all in preclinical models of disease. So it's really hard to connect our behaviors of interest to changes in physiology. The way we try to tackle this in the lab is we're essentially trying to combine that cardiology approach of measuring electrophysiological activity with behavior. Behavior in the case of heart disease is your heart pumping blood to your brain. Behavior in the case of the brain is a broader range of activity. And we do this by implanting electrodes into the brains of animals. So instead of an EKG, which is measured from the surface of your chest, we are measuring electrical activity from deep within the brain, from many parts of the brain simultaneously. So rather than being a 12-lead EKG, this is a 12 to 16 lead within brain electrical signature. And that electrical activity, like the EKG signatures, we're able to process them, and we're able to use engineering approaches to process these online. Each activity, it's like a brainwave. So like the EKG or like the scalp electrodes you put on the case of trying to measure seizure activity, it's a wave of activity, and we can look at how much activity is distributed across different frequencies. In other words, is there activity that's happening 1 time a second? This might be 60 beats per minute, and things that are happening faster. So we can process those electrical signals at each lead of the brain recording technique. We're also able to use engineering principles, which suggest that things that change together across time tend to lie within the same system. So we can ask if those different leads of the electrodes in the animal's brain are changing together, if the electrical activity changing together across time. And we term this coherence or synchrony. Once we're able to get these electrical activity, we can also ask if these electrical activities are changing between regions in a way that's patterned. For example, we can do mathematical tricks and ask if activity in area A predicts future activity in area B. And if it is, that means the electrical activity tends to be moving from area A to area B in the same way electrical activity moves from your atria to your ventricles. We can also shift it in the other direction and infer whether information is moving from area B to area A. So as you can imagine, from these many, many leads of electrodes in an animal's brain, we can ask what's going on in each area, what's going on between areas, and how this changes across a range of frequencies. You suddenly have a big data project. And so much of what I'm going to spend the remainder of the talk out here is explaining to you how big data connects with neuroscience, connects with psychiatry to move us towards precision diagnostics. And this work was led by Kyle when he was a graduate student in Larry Curran's lab, David Carlson as well, who's now on faculty here in the medical school, and Neil Gallagher, who's a graduate student in my lab. And what they were able to do was ask this question: if you have all of this electrical activity, can you find patterns in there that make sense and relate to behaviors of interest to psychiatry? And this is an example of the model they came up with. It's one of the first ones we have in the lab. We've since developed many more, but it's a 3-tiered model, making it hierarchical. So there are 3 layers. The bottom layer is our neuroscience level, which says we are measuring electrical activity that's really fast. It's changing on the order of milliseconds, so thousands of times per second. And we can measure that for many brain areas, and we can measure it and look at activity across frequencies, as well as how brain areas are synchronizing together. So for each animal across time, we're getting thousands and thousands and thousands of observations or measures. We then apply the engineering principle that says things that change together across time tend to be related, and that's our second layer. And we're going to say emotions, which are changing on the time scale of seconds. In other words, we're saying you're going to be happy for seconds or sad for seconds. None of my patients has ever been complained about being sad for a quarter of a second. We're going to say emotions are linked to brain processes that change together at the second's time scale. And then happiness might be process 1 plus a sprinkle of process 2, process 6 plus a sprinkle of process 1. And what our machine learning algorithms are going to do, machine learning just takes all that data and finds patterns. It's going to learn where the red lines are. So I'm going to show you an example on the next slide of what this brain EKG looks like. And the paradigm that we're going to do this in is across social behavior. So we pick social behavior because fundamentally what psychiatric disorders do is they impair the ability of human beings to interact with each other. And this shows up either in your daily life or in your occupational function as well. So experimentally, we're going to put electrodes into this mouse, and then we're just going to let it explore an arena for 10 minutes. And in that arena, it could spend time with another mouse, or it can spend time with an object. And it can basically run back and forth and spend its time in between these 2 scenarios. We're able to do video tracking, and we record brain activity as the animal is doing this. And then we do this across 10 days. Each time the animal gets 10 minutes to spend time with a new animal or a new object. We record brain electrical activity from many brain areas, both on the surface of the brain and deep within the brain. And then we put it through our machine learning algorithm to say, what does the brain network or brain EKG signature look like? So this is what we get. These are each of the brain areas that we've recorded. Several areas on the surface of the brain, several areas deep within the brain, like amygdala, and part of the brain called the nucleus accumbens or reward circuitry. What you see around the area from here from 1 to 60, this is frequency. So 60 here means basically once, 60 times per second. The 1 here means 1 time per second. So imagine this is 60 beats per minute or 1 beat per second. And this is what's going on within each brain area. If there's color there, it means activity in that brain area, in that frequency, contributes to the EKG signature. And if you see a line between areas, it means these two areas tend to synchronize with each other. So each area, think of it as an instrument. Each instrument can play a sound pitch or a tone, and then the conductor causes the areas to play together to play music. And we're also able to track electrical activity as it's flowing through all of these brain areas. So machine learning gives us this complex brain-like EKG map. And then what we're going to do is ask the question, so now we've got this EKG signature of when animals are socially interacting or interacting with an object. Does this work on a new animal? In other words, if a new mouse comes into our mouse emergency room, does this signature also tell us what's going on in their brain? So we take 28 new mice, implant with electrodes, and then we let them socially interact. And here you see the time where a social interaction starts. You see the time where a social interaction ends here. And then in green, it's what this brain network or this signature looks like as an animal starts interacting and when it stops interacting with another mouse in green and another object in red. And as you can see, this network that's related to social behavior basically turns on and tracks when an animal is socially interacting with another one, and it tracks exactly when that social interaction is terminated. So we have a brain-like EKG signature of social behavior. Importantly, and we always say this is an important feature of everything we do, we make sure that the signature works in both male and female mice. And another feature of this, if I'm just showing you how well its brain can tell the difference between a social situation or an object situation, 0.5 means no information. The higher it goes, the more of this network is useful. Each of these is an individual mouse. What we show is that the more an animal prefers to social interact, so the more it chooses to spend time with another mouse, which we are using as an interpretation of its desire to socially interact. So the more the animal likes or enjoys socially interacting, the more its brain can tell the difference or decode the difference between the social situation and the object situation. In other words, we have found a network that tells us how much animals like to interact with other animals. And so we got really excited about this because of course there's psychiatric disorders where there's disturbance of the ability of individuals to enjoy social interactions. And so what we're going to do now is we've got this network, we know it works in new animals, we can tell how it's related to biology and brain areas, and we know it works in new contexts. In other words, we can show how robust this signature, this brain signature of social interaction is. So we then wanted to ask the simple question, does this network fail to encode properly in animals that we know are reasonable animal models of autism spectrum disorder? And as you know, autism is a neurodevelopmental disorder that is affecting 1 out of 50 to 60 young people in this country. And this disorder is associated with profound deficits in social behavior, communication, and repetitive behaviors as well. And there a new postdoc in my lab who was recently in Van Bennett's lab for graduate work, they were able to find what was a risk gene. In other words, having this gene, a mutation in this gene, gives you high risk for autism. They found a mutation in a human subject, put it in a mouse, and then created a mouse line that expressed this human gene which gives risk for autism. And we wanted to ask the question, does this risk gene in humans change the way this social network encodes? In other words, does this risk gene change the brain EKG signature? Now, we knew when we put these animals in this social assay, which asked how much time they choose to spend around a social another mouse or another object, there were no differences in their grand social behavior. And historically, one would throw this out and say, well, it's not a good mouse model of autism. But having spent some time in an autism clinic during my residency training, I realized that many of the patients I was taking care of would choose to spend time really close to me, even though their brain was not entirely processing the social information the same way. So when we put electrodes in these animals' brains and we asked how much this brain EKG for social behavior was encoding their behavior, we found in the wild type littermate controls that relationship was still there. So the more a mouse chose to socially interact, the more their brain encoded the social condition. In the mice that were models of autism, we found that this relationship was totally disrupted. In other words, the way a normal animal's brain encoded social information did not extrapolate to the animals that were a genetic model of autism. And then the final study that we did this in conjunction with Karina Block, who's a graduate student in psychoneuro, and my collaborator, Stacy Bilbo, who's in the same department, and Chala, who's in the Department of Cellular Biology. We took a mouse model that they'd worked on creating, and it's based on exposing animals to a stressor when they're in their mother's womb. And so they both get a stressor, an environmental stressor, in terms of the bedding being changed in the mother's cage, as well as an environmental pollutant, in this case, diesel particle, which is thought to activate the immune system of the mother while the young pup is in the womb. And this has also been used as a model of autism. And so they put the mice through this protocol. We waited for them to grow up, and then we put them through the same assay. And when we did this, interestingly enough, autism, which is much more penetrant in males than females, we found that the male mice, and not the female mice, just the male mice, showed differences in their social behavior. Interestingly enough, these animals tended to spend more time around another mouse. And again, historically, one might throw this out and say, well, it's not a good model of autism. But we were interested in this fundamental question: does this manipulation change how the brain EKG is encoding the social information? And when we do this, on the left, I'm showing you the male mice. On the right, I'm showing you the female mice. In the black, I'm showing you the healthy controls. In the red, I'm showing the animals with the manipulation. So I'll start with the females. In the female mice, both the animals that did not have the early life manipulation showed normal coding in this network. And so did the female mice that had been exposed to this stressor in their mother's womb. On the other hand, the males, the normal animals, showed normal encoding within this network, the relationship between social behavior and encoding. But the animals, the male animals that had been exposed in their mother's womb, this relationship was disrupted. And it was disrupted in the same way it had been disrupted in the Ankyrin 2 mutants. So I think this type of research opens up the avenue to is our ability to begin to understand what these network level patterns, these brain EKG signatures look like in the animal's brain to develop precision diagnostics. So I started off with this slide. And our vision for how this field will go forward is to begin to learn independent networks for a range of behaviors that we can then port from mice into humans. And this is a picture of my lab. It's not the most recent one, but it just shows the individuals who've gone forward doing this work. So thank you all for having me here, and I'm happy to stay around for any questions at the end.